Hello everyone, welcome back to my channel. Today we are again diving into general histology. So part 1 is already uploaded in the general histology series section. Let's get started. So this presentation is a continuation of that first part. In the first part we talked about the plasma membrane. Now let's talk about the cytoplasm. Cytoplasm is like a bustling city, we can say, inside a cell. Why is that so? Because it contains all the necessary organelles to keep the cell alive. For example, it has mitochondria for energy reserve. It has ribosome, which is for the protein synthesis. So all of these organelles. Additionally, it also has cytoplasmic inclusions, which we call as metaplasm. For example, glycogen. Glycogen is for energy reserve. Then pigment granules like melanin. Then we have secretory granules in which there will be substances which are ready to be released. Hence, it is called secretory granules. Then there is lipid droplets. It also has cytoplasmic matrix, which is called as cytosol. And lastly, it has ground substances. Let's first talk about the membrane bound organelles of the cytoplasm. So the first one is rough endoplasmic reticulum, commonly called as RER. It is a site of protein synthesis for export outside the cell. And this rough endoplasmic reticulum, as we can see in the diagram, they are studded with ribosomes. The second is smooth endoplasmic reticulum. Smooth is for steroid synthesis in adrenal cortex and testes. If we talk about cardiac and skeletal muscle, so their smooth endoplasmic reticulum is for sequestering calcium. If we talk about liver, so there the function of smooth endoplasmic reticulum is for lipid and glycogen metabolism. So remember, in different organs, smooth endoplasmic reticulum have different function. So let's talk about Golgi apparatus. We can remember Golgi apparatus like a shipping department, like a post office. There are two ends of Golgi apparatus. First is cis phase, another is trans phase. So in the cis phase, there will be incoming proteins from the rough endoplasmic reticulum. So RER will synthesize proteins, and through cis phase, those proteins will be sent to Golgi apparatus for its modification and packaging. Now, what is this modification? So basically carbohydrates will be added to these proteins, and through the trans phase this will be sent further to be released. So that's how it is called as a post office or shipping department. Along with that, there is also lysosome production in Golgi apparatus. Now, talking about mitochondria. We all know there is energy production in mitochondria. So, ATP production via the Krebs cycle and oxidative phosphorylation, and it is not present in RBC. Very important line, mitochondria are absent in RBC. Let's talk about the cleanup crew of the cell. First, let's talk about endosomes. Endosome basically is like a transport system. So whatever vesicles are formed as a result of phagocytosis, endosome will transport that vesicle to lysosome for further digestion. Lysosome with their hydrolytic enzyme, remember hydrolytic enzymes, they will digest whatever microorganism or cellular debris to clean up the cell. Lysosomes are produced by Golgi apparatus. Then lastly we have peroxisomes. Now peroxisome is very important because through the oxidative enzymes like catalase and peroxidase, it will digest hydrogen peroxide, which is very harmful to our body. And hydrogen peroxide is a byproduct of metabolism. So that's why all these organelles, lysosome, peroxisome, endosomes, we call it as a cleanup crew of the cell. So question, which of the following structure is involved in sequestration of calcium in cardiac as well as skeletal muscle? So RER, that is for the protein synthesis. Peroxisome, for digestion of hydrogen peroxide. Then smooth endoplasmic reticulum, yes, it is responsible for sequestration of calcium in cardiac as well as skeletal muscle. And lysosome, it is responsible for digestion of microorganism through hydrolytic enzyme. So the correct answer here is option C. So let's talk about the non-membrane bound organelles. So first is microtubules. Microtubule provide cytoskeletal support. It is for intracellular transport and cellular movement. They are composed of tubulin. Remember, tubule is for support, transport and movement of the cell. Then we have axoneme. Axoneme are nothing but specialized group of microtubules. They are found in cilia and flagella of bacteria. And the cilia and flagella, we know they are responsible for movement of bacteria. That is for basically locomotion. Then we have centrioles. Centriole is the one which provide microtubule organization. They forms the end of mitotic spindle during mitosis. Remember, this is important, that centriole forms the end of mitotic spindles. Then we have microfilaments. So these are nothing but our actin and myosin, which we know that it is responsible for muscle contraction, and this also provide cellular movement or support. Then we have intermediate filaments like vimentin, desmin, cytokeratin. So basically, vimentin is found in connective tissues for cytoskeletal support, and desmin is found in skeletal muscle fiber for its support. And cytokeratin is also responsible for cytoskeletal support, and it is found in the epithelial cells. Then we have basal bodies, which are derived from centrioles, and they are responsible for development of cilia. So when we talked about RER, we had said that RER is responsible for protein synthesis to be used outside of the cell. But ribosome is responsible for protein synthesis for use within the cell. And this contains rRNA and proteins. Let's quickly go through the nucleus. Now, nucleus also has a membrane surrounding it, and this membrane is made up of inner and outer plasma membrane. Then, similar to cytoplasm, nucleus also has nucleoplasm, which are ground substance of nucleus. So, cell's ground substance is cytoplasm, nucleus ground substance is nucleoplasm. Nucleus has chromatin, which is nothing but complex of DNA and protein, basically histone protein. Now, whenever this chromatin are loosely arranged, it indicates that there is nuclear activity going on. Remember, euchromatin indicates nuclear activity. These are loosely arranged chromatin. However, when there are highly condensed chromatin, then they are heterochromatin, and that indicates that there is no nuclear activity going on. Remember, highly, H is for highly condensed, H is for heterochromatin. Now, nucleolus, very important because it is a site of rRNA synthesis. rRNA is responsible for protein synthesis. So remember, very important thing, nucleolus is non-membranous. There is no membrane surrounding nucleolus. Bare body, it's not that important. It's just a repressed X chromosome found in the cells of females, and it appears as a dense chromatin mass. Its presence is used for the sex identification because it's found only in the female. So question, transcriptionally inactive chromatin is termed as prechromatin, euchromatin, heterochromatin, or none of the above. We just talked about it, that H is for highly condensed, H is for heterochromatin, and whenever there is highly condensed chromatin or heterochromatin, it indicates that there is no nuclear activity going on. So the correct answer here is heterochromatin. Now, what are the structures which are present on the surface of cell? So first, microvilli. These are finger-like structures and they can be of various length. Especially they are located on the apical surface of epithelial cells. And these are composed of microfilaments. Provide increased surface area for absorption and transport of fluid. Second is stereocilia. These are usually long microvilli. They are located in epididymis and sensory cells of inner ear. Then we have cilia. Cilia are nothing but they are short hair-like structures, and these are for the locomotion of the cell. They function in a synchronous and coordinated wave-like motion. The cell body produces cilia. Flagella. So cilia, if it is a short hair-like structure, flagella, they are long whip-like structure and they are also for locomotion. They function in a snake-like motion, and humans, they are only found in spermatozoa. So that's all for the cellular structure. Do check out the first part for plasma membrane, and see you all in next video. Till then, happy studying, all the best. And if you like this video, please like, share and subscribe to my channel. And do visit the head and neck anatomy series also. There are many videos uploaded there. And also let me know if you want any specific topic to be covered. Till then, happy studying. Bye.